suppose that we are crossing with a tall plant, tall pea plant. It may be male or female, whatever. So for the tall, I told you that the genotype will be capital T, and a short plant, mean dwarf plant, right? And that will be the small t. Genotype genes will be factor will be small t. Then I said that these are the parental generation. Remember, these are the parental generation. In parental generation, what will happen? First, the formation of gamete will take place. When formation of gamete will take place, both of the genes, so genes are found in pair, they will just separate out, right? The genes will get separate out. When what I said, so this is the gamete formation. It is also called gametogenesis, means gamete formation. So gamete formation taken place. Gamete means sperm and egg formation will take place. Then I told you, if you remember, then I told you that what will happen, the cross between two will take place. When the cross between two will take place, how we are going to make the cross? Just a minute. Look, how the cross will take place. Actually, I'm going to write it down. Just a minute. Right, I got it. Okay. So what I said to you that I said to you that I said that there will be two crosses. So if we cross a tall plant with the short one, right, short or dwarf. So for the tall plant, genotype will be, I said that let's take the genotype capital T, and for short plant small t. What happens? So we call it parental generation. Parental. And please let me know if you are unable to understand it. Then what will happen? First, the formation of gamete will take place. So how we will make the gamete? Gametes. So genes are always found in the pair. We discussed it on in the last class. So capital T and capital T. Then I said for the small t and small t, both of the chromosome get separated during the time of what? Gamete formation. So these are the gamete. Then I told you how the cross will take place. So for the sake of the cross, what we are going to do, we just what you have to do, because you can because it can come in your examination, right? So first we have to take this one and we have to multiply with this one. Then what will happen? Capital T, small t, right? Then again we have to take this capital T and we have to cross with this another t, right? Now what will happen? Capital T, small t. Then what we have to do? We have to take this t and cross with this one. Then what will the combination? Capital T, small t. Again we have to take this t and cross with this small t. What will happen? Formation of capital T, small t. So that's how the formation will take place. And Mendel have seen that in the F1 generation, so whatever we see, right, whatever we see with the naked eye, that is called phenotype, means physical characteristic. So he seen that all plant are tall plant, right? So all plant are tall, right? Here the this capital T means the gene for the tall. It is able to reflect itself, it is able to represent itself, while the effect of a small t is hidden over there. So he said that in F1 generation, we call it filial one generation, F1 generation. The first generation means F1 generation. in first generation, he said, when we will cross with the tall pea plant with the short pea plant, what will happen? That all the plant will in F1 generation, all the plant will tall, right? So what happened? It means that the gene in F1 generation, which is able to represent itself, which is able to express itself, it will be called dominant gene. What we will call? We will call dominant gene, right? And the gene which is unable to show it, right, which is unable to represent it, right, unable to express itself in front of capital T, that will be called recessive gene. This is called law of dominance. Now what is the phenotypic ratio? Phenotypic ratio. So what a phenotypic ratio means? What we can see, physical characteristic. So how many shots are? 0. And how many tall are? 4, right? What is the genotype? Genotype means genetical characteristic. All are no even single plant is small t or capital T, all capital T small t. So phenotype is also 0, 4, means all tall, all heterozygous, right? So is it clear or not? Please tell me. Is this
Yes,
process
sir.
clear? Please note it down first if you haven't. By the way, I have told this genotype, this process in last class as well. Please note it down fast. Done?
Yes, sir. Phenotypes like the first generation.
F1. This is called filial one generation. F1. Right? So please note it down. The gene which express itself, the gene which express itself even in presence of, even in presence of alternative gene, even in presence of alternative gene will be called dominant. Full stop. It dominate, it dominate the recessive characteristic, it dominate the recessive characteristic on other pair. full stop, the gene which is unable to, the gene which is unable to express itself, the gene which is unable to express itself in presence of alternative gene is called recessive gene. One more thing which I want to share with you, if the genes are similar, both pair are similar, capital T or small t, these kind of the pair is termed homozygous. But if there is the gene like this way, one individual is another and another individual is different, then we call it heterozygous. So actually dominant gene presents itself in homozygous condition as well as in heterozygous condition, but recessive can represent itself only in homozygous condition, right? So please note it down. When both pair are similar, it is called homozygous. When both pair are different, it is called heterozygous situation. Now what else? One more important thing here, what I want to share with you. Now what we can say, take one individual, take two individual for F1 and do the cross between them. This is called shelving. So what we taken a capital T small t pair, right? We have taken from here, right? And one more capital T small t. This is called shelving because we are making the cross between the F1 individual, right? So this is called shelving, right? So we did this shelving. Again, what we'll do, the next generation will be F2 generation. What we will do, we will make the gamete. How the gamete formation will take place? Look, capital T, small t. Again, capital T, small t, right? So now again we will make the cross. Let me make the cross. So when we make the cross, we will take this capital T and capital T. Now this will be what we will call, this is called homozygous tall, right? This is called homozygous tall. Now what will happen again? What will happen? Capital T, capital T, small t. Now what is this? This is heterozygous tall. So what is this? Is tall, but how which kind of the tall? Homozygous. Homozygous, right? This will be, again it will be tall, phenotypically tall, but what is the genotype? It is heterozygous. Now again this capital T, this small t will cross with the capital T. What will the capital T small t? What will the phenotype? It will look like tall plant. And which kind of the gene is? Heterozygous. Again, this small t will cross with the small t. Now what will happen? Small t. What will be? This will be short plant. And what kind of the gene? Homozygous. So I said that recessive gene will represent itself in only homozygous condition. So what is the phenotypic ratio? What is phenotypic ratio? When we see phenotypic ratio, so there are 3 tall and 1 short. So phenotypic in the look, morphology, so the ratio will be 3:1, right? What is the 3:1? 3:1 means ratio of tall versus short plant, right? Now what is genotypic ratio? So what is genotypic ratio? A homozygous tall 1, then so 1 homozygous tall, then 2 heterozygous tall is to 2, right? And then 1 homozygous short into 1. So this is the ratio of genotypic ratio. So in F2 generation, and this will be called F2 generation, filial 2 generation, right? In F2 generation, the ratio will be phenotypic ratio 3:1, tall is to short, and genotypic ratio 1:2:1. Is that clear or not? Please tell me. Is that clear? And if it's clear, please note it down quickly. This is called law of dominance. Done. Martin, done.
No, no, I'm doing.
Okay. They can ask you for this cross, monohybrid cross, and I have already told you in last class. Monohybrid when single characteristic is taken, dihybrid when two characteristics are taken. In any cross, if single characteristic, like if we have taken only length, here we have taken only length, this is called monohybrid cross. If we take the length and color of flower, that means we have taken two characteristics, then that will be called dihybrid cross. Let me know when you complete it, when you note it down.
So genotype means?
Genotype means genetical characteristic, genes, the composition of genes, right?
Yes, sir.
So genes are capital T, capital T. This is the genotype, right? Means genetical constitution. Phenotype means external characteristic, morphological characteristic. Amna, is it clear to you? Martin, should we go ahead?
Yeah, yes.
Okay, fine. So this was the monohybrid cross, right? Mendel's monohybrid cross, because we are here, we have taken only one characteristic, that is the length, right? Now, this was the Mendel's first law, law of dominance. Then I'm going to tell you that Mendel's second law. What is the Mendel's second law? Mendel's second law is law of segregation. What is the law of segregation? Look, what Mendel says. Look, here you can see easily that this capital T and capital T, right? This capital T, small t. These both of the gamete, they are found in pair, like in me, in you, both act like in me, X and Y is present, right? When they will convert in the form of gamete, they will separate, X will separate and Y will separate, right? Look, in the same way, capital T, genes are found in pair, right? When during the gamete formation, right? Means gametogenesis, during the gamete formation, they separate. Segregation means separate, right? They do not. So what happened? The gamete formation will take place. Again, the formation of capital T. Look, you can see here the formation of capital T, small t taking place. These capital T, small t do not intermix with each other. Genes will not intermix. Again, what happened? Again, in F2 generation, this is the F2 generation, right? And this was the F1 generation, right? During F2 generation, again, what happened? Formation of gamete taking place and genes separate, right? And so during the gamete formation, what is the law of segregation? According to law of segregation, genes or factor, Mendel do not aware about the genes. He used the term factor. So he said factor do not intermix each other, rather than they get separated from each other during the gamete formation process. Is that clear? This is called law of segregation. Is it clear or not? Please tell me. Right?
Yes, sir.
Please note it down. Law of segregation. Law of segregation. The same cross will applicable for the law of segregation. According to this law, factors are found in pair. Factors are found in pair, but they do not segregate. Sorry, the factors are found in pair, but they do not intermix. Factors are found in pair, but they do not intermix. Full stop. At the time of gamete formation, these factors get separated. From each other and then passes to next generation. This is called law of segregation. This is called law of segregation. Full stop. You can, for example, capital T, small t do not intermix. At the time of gamete formation, they separate from each other. This was the law of segregation. Is that clear?
Wait, so like T capital T small t separates.
Sorry?
Say separate, like it's become T capital T small t.
Capital T and a small t separate from each other and then passes to next generation. Look, now there is a third law of Mendel. Law of independent assortment. I told you in last class, law of independent assortment. What is the law of independent assortment? Law of independent assortment is every factor. Look, first they are found in pair. Then what is the law of? Then what is the law of segregation? Both get segregated. Now this capital T here you can see this capital T. Look, this capital T is free to go with this one, either this one, wherever it want. It is there is a no compulsory. There is no compulsion that this capital T will always remains with this capital small t. No, no, no. This capital T can go with this one or either this one or this one. Wherever it want to go, it can go. Right? There is no compulsion. Right? So this capital T can cross with. That's why we cross like this way. This capital T is free to go with this one. This capital T. It is also free to go with this capital T. So this is called. It is independent to go with any other gene, right? To make pair with any other gene. This is called law of independent assortment. Same way, like this is small t, it is free to go with this capital T and also free to go with this small t, right? That's why we cross with the both of them, right? So every gene is independent to go with another gene, right? This is called law of independent assortment. Is it clear?
Can you repeat, sir?
Yeah, sure. Look, here, this capital T, we cross this capital T with this one. We also cross with this one. It means these both are genes are with each other, right? Look, this both capital T are with each other, but there is not any compulsion with this capital T that it will always remains with this another T. This capital T gene can go with this small t, or it can go with this small t. It is free, it is independent to go with any of these two, right? Any of these two, right? Any of the genes. There is no compulsion. These both capital T will always remains with each other, right? This is called independent assortment. Is that clear?
Yes, sir.
For example, that you and your friend, you always remains with each other in the classroom, but this is you always remains with each other in the classroom in a pair, but there is a no compulsion that you will always sit in the class with your best friend, right? Sometime you can sit with the backbencher, sometime you can sit with the middle bencher. Wherever there is no compulsion of both of you, neither you complain with your friend nor your friend complain with you, right? Even the same your friend is free to go with any another friend, right? So that is called law of independent assortment. It means you both live in the pair, you both remains in the pair, but there is a law between you that you have make an agreement that is independent assortment. Any of you can go with any another person, right? In the same way with the genes, genes always remain in the pair, but they are free to go with any another gene during the cross, right? Is it clear? Is that clear? Amna, is that clear?
Yes, sir.
Right. So please note it down. Law of independent assortment, the third one. Law of independent assortment. Look, please note it down. According to this law, law of independent assortment. According to this law, the factors are free. The factor are free to go with any other factor, to go with any other factor during the cross. The factor means genes, right? Because Mendel do not know about the genes, he used the term factor, right? Factors are free to go with another factor during the cross. This is called law of independent assortment. Right? So look. Now, what is the second in your syllabus? Dihybrid crosses given in the syllabus. Then sex determination, right? Then the next thing which is in your syllabus, that is a dihybrid cross. I have told you that dihybrid cross means two characteristic we will take, two characteristic with the cross. Dihybrid cross. Give me 1 second. Okay, and right. Look, I already told you that law dihybrid cross means when we take two characteristic. Look, there we taken only one characteristic, that is the height of the plant, right? Here, this is inheritance of two gene. When we take two characteristic, what two characteristic here we taken? The same thing is in your textbook, right? So here we have taken two kind of the first characteristic. What we taken, that is a coat, seed coat of shape of the coat, seed coat. So the seed coat here is first shape is, it is round, it is wrinkled. So first we taken one characteristic, that is a seed shape. So seed shape here we taken round and wrinkled. For round we have taken capital R, capital R. For wrinkle we have taken a small r, right? Again, so this is the one characteristic. Then in dihybrid cross we have to take two characteristics, means two quality. Here the second thing we have taken the seed coat color. This is yellow and this is what? Green, right? Again the color is yellow. For the yellow, genes is capital Y, and for the green the small y, small y. Is that clear? So here we are taking two characteristics: shape of this shape of the seed coat and the color of the seed coat, right? What? P. So parental generation, the gamete will be, what will be? There will be two pair, capital Y, capital R, capital R. So capital Y represent what? Capital Y represent yellow color and capital R represent round shape. Here small y, small y, small r. Here small y represent green color and small r wrinkle shape, right? When the gamete formation will take place, what will happen? How to make the gamete? This capital Y, we will take capital Y, capital R, right? So capital Y will be first gamete. Here you'll make a mistake. Look, what we will do first, we will make the gamete. How we will make the gamete? While making the gamete, this capital Y, capital R, capital R. First, what you have to do? First, what you are going to do? First, you have to take this Y, cross will this R. So combination will be capital Y, capital. Every time the combination will be same. Then you will take this capital Y, this R, capital Y, capital R. Again, what you will do? Take this Y, this R, capital Y, capital R. Again, this Y with this R, capital Y, capital R. So every time you will make the gamete capital Y, capital R. That's why during the gamete formation we have taken capital Y, capital R. Is it clear? This first step, it is very important. Here you will make the mistake. Is that clear, Martin?
You do it. Yeah.
Okay. Now again, Amna, is it clear? Again the same. Capital Y, small y, small r. Same. You will cross whatever you will cross. Your answer will be small y, small r. Again, small y, small r. Again, small y, small r. Again, small y, small r. So the same what when we form the gamete in this combination here taken capital Y, capital R, and then capital small y, small r. This is the parental generation. Then F1 generation. All the plant will be what? Capital Y, capital R, small r. So dominant gene is yellow and dominant gene is round. So all plant are what? All plant are round and yellow. So what will be the genotypic ratio? What for phenotypic ratio? 4:0. Is it clear? Tell me, is it clear or not? Tell me, guys.
Yes, sir. Yeah, yes, sir.
First, please note it down. Then we will go for the shelving. Done, Martin?
Yeah, no, I'm doing. Should I draw a table?
Yeah, we have to draw the table, but how we have to draw the table, I'll tell you, because that I have to tell you. Be a bit faster. Else. So when you have your final examinations, Pardon?
Fourth, I think. For
Fourth,
science, for science.
first,
First is language, 20th.
first April or March? April.
March, sorry, February. Starts first exam at February 20.
First February means it's going on.
No, it's going to start.
Going to start from first of
No, 20th of February.
20th of February. Okay. Because we are left with one more chapter, that is our environment. Yes.
Yeah.
Fine. Done. Look. Now, now you'll be surprised that how come this capital Y capital R. Then again, what we have to make? Then we have to again make the gamete, right? Why? Because we have to uh, uh, we have to do the shelving, what we did in last cross, right? So when you will make the gamete of this one, look how the gamete will be made. Capital Y, small y, capital R, smaller. That is. So how the gamete will be made? Gamete will be made like this way. First, we will cross this Y will capital R, right? Capital Y and capital R. That's what we put over here. Look, again, this capital Y will be crossed with this small r. Capital Y, small r. That's what is put over here, right? Then again, this small I, Y will be crossed with capital R. So what will be? Capital R and small y. That's what we put. Capital Y, small r. Yes. What is small? Sorry, capital R, small y. Capital R, small y. Yes, that's what we put here. Then again, this small y with small r. That's what we put here. And the same thing, same gamete are put over here, right? Here, in the form of OA, here in the form of sperm. Then what we did, we crossed with this one. Capital I, this capital Y, capital R will be crossed with this one. Again, this capital Y, capital R will cross with this one. Then again, this will cross with this one. This will cross with this one. Whatever the outcome, so when we cross this one with this one, outcome will be here. So capital Y, capital R cross with capital Y, capital R. We written over here. Again, this capital Y, capital R is crossed with this one. Capital I, capital Y, capital Y, capital R, small r, right? Again, this capital Y, capital R crossed with this one. Capital Y, small y, capital R, small r. Again, this capital Y, capital R crossed with this one. Outcome is here. Now, on the basis of the gene, look here, both of the gene, two dominant gene are capital Y is present, one capital Y is present, so it represents yellow color. One capital R is present means round color, right? It will be yellow round, right? Again, this will be we will repeat the same thing. This will be first cross with this one, we will put here. This will be crossed with this one, we will put here. We will cross this one, we'll put here. We'll cross this one, we'll put here. That when we will make all the crosses, what will to total offspring? So to total offspring of 16. Out of this 16, 9 will be yellow round, that is dominant phenotype. 3 will be out of 16, 3 will be green round. Again, 3 will be yellow wrinkled, and only one combination will be green wrinkled. So in dihybrid cross, the F1 ratio was, phenotypic ratio was 4:0, and the phenotypic ratio, here we do not count genotypic ratio. Here phenotypic ratio in F2 generation in dihybrid cord 9:3:3:1. Tell me, is it clear or not?
Yes, sir.
So that's what you have to note it down. If you want, you can take the screenshot and you can note down after the class as well. There is no issue. But you have to learn, where you will make the mistake, you will make the mistake, prime mistake. The normally student make mistake here at this point. You make this one. And tell me, Hamna, is it clear or not? Did you got all things or not? Say frankly. It's a bit complex, but it's not too complex.